Hello, dear colleagues. This is a short introductory course in Biostatistics. The course is designed for statistical programmers, MS and PhD students, and biomedical researchers. No statistical knowledge is required for the audience except for some basic knowledge of algebra. I have to apologize for my English. As you have noticed, I'm not a native speaker. However, this shouldn't stop us. The goal of this course is not to study statistics, but rather explain some basic concepts and ideas behind descriptive statistics and statistical analysis. In order to give you deep insights into these concepts and avoid mathematics, large number of visualizations will be used. There will be 4 lectures. We will start from introduction to different types of mistakes, errors, that one can encounter while conducting a study. We will find out which of these errors might be controlled by statistics and which might not. Next, we will talk about descriptive statistics and pay special attention to normal distribution that we will use then for testing inferences. The final lecture will give you some hints on how inferential statistics works. You will learn about the principles of construction of confidence intervals as well as calculation of p-values. For complete understanding of these paradigms, you will learn how to perform a simple one-sided t-test, also known as student's test. On this slide I list the textbooks that were used in preparation of this short course. Among them, Primer of Biostatistics by Stanton Glantz is one that is written for non-statisticians and is easy to read. Other 3 books are intended for master degree students. Today we will start from epidemiology. In simple terms, epidemiology is the science that studies causality of various diseases and health states. Back in time, evidence-based medicine originated as a branch of epidemiology. Epidemiology is essential for statistical thinking because statistical tests are used to identify cause and effect associations, and not just any associations. One of the important topics in epidemiology is a doctrine about the types of mistakes that one can make during a biomedical research. Some of these mistakes can be controlled by statistics, but most of them not. By studying these mistakes, we will learn not to overestimate the power of statistics. The first concept is validity. Validity is the ability of your study to provide you with adequate measurements. It is your confidence that your measurements reflect the essence of the phenomenon that is under investigation. For example, it is valid to use a medical thermometer to measure body temperature. However, such thermometer will give you invalid results when measuring room temperature. I was told of a thesis where a student used a psychological questionnaire, MMPI, to predict atrial flutter in elderly subjects. The questionnaire was designed to describe such psychological traits as depression, hysteria, psychopathy, and so on. These questions had no relation to the somatic cardiac diseases, yet the student got a significant p-value for a combination of psychological scores in subjects with and without atrial flutter. It is quite obvious that any psychological questionnaire is invalid for screening for cardiac diseases, and the student got a significant p-value because of some incorrect calculations. The question is, do we need to check statistical calculations when we know that the study lacks construct validity? The answer is no, there is no need to understand where miscalculations took place. Invalid research automatically goes into the trash bin. The next concept is random errors. Let us imagine a shooter trying to hit a target. First time he shoots from a short distance and the spread of hits is small. Next time he shoots from a long distance, the spread increases. Though there is not a single hit exactly in the center, we can guess that the shooter aimed at the center. Such fluctuations are called random errors. This is the main source of errors that statistics deals with. Let us imagine that there is some problem with the rifle. The shooter aims at the center, but the hits go to the right. We have got two sources of error now. Random errors provide the same spread. A systematic error adds a constant right decline. Systematic errors are also called bias. Bias cannot be controlled by statistics, and biased data automatically go to the trash bin. Although various techniques have been developed to deal with biased data, they are not used in clinical trials. Let us look at how statistics helps us to control random errors. We might want to calculate the mean for the hits to understand what was the aim of the shooter. Statistics provides us with the tools to calculate the probabilistic limits for the mean, known as confidence interval or confidence region, that will reveal the most probable area on the target the shooter was trying to hit. Here we have the confidence interval for the horizontal spread of the bullets. Since our shooter was aiming at the center, confidence interval must include the center. However, it cannot include the center if we have a systematic error. In this case, we make a wrong conclusion about the shooter's true intention, and statistics doesn't help. Our biased conclusion may be statistically significant, nevertheless it remains wrong. It is quite common to get the mean that is different from the true value because of prominent random errors. Still, the confidence interval will compensate for the wide spread and will include the center, the true aim of the shooter, but not if there were some problems with the rifle. You have probably guessed by now that by the center of the target I imply the population mean, which we are trying to estimate with the sample mean. Thank you for your attention. We will proceed in the next section.